Good morning, everyone. Am I audible?
ઉઠાડ્યો મેં અને તો નથી ના જો સવારે મેં 4-5 વાર ઉઠાડ્યો એટલો નસકોરા બોલે છે એના એ છે ને એકચ્યુઅલી નસકોરા છે કે એના નાકમાં શ્વાસ લે છે એમાંથી અવાજ આવે
Hello. Good morning, everyone. I'm going to start today's session. I welcome you all and everyone. Please be comfortable. But I want to know, am I audible?
Yes, ma'am.
Yes, ma'am.
Okay. So first of all, I would like to wish a very happy International Microorganism Day 2023 to everyone. As you all know, on 17 September, it is considered as International Microorganism Day. So we are very delighted that on this special day, we have with us Madam Dr. Sunita Satish, ma'am. So I welcome you, ma'am. Ma'am, are you there? Sunita, ma'am, are you there? Ma'am is with us now. Let me introduce myself first. Myself, Saida Tamanna Yasmin, State Coordinator, Microbiology Society India from Assam, a district microbiologist, Saradio, a district of Assam, and a research scholar microbiology. So it is immense pleasure of mine to introduce today's guest with you all. Uh, as I said, we have Dr. Sunita Satish ma'am. So she is presently head of the department microbiology and coordinator of science faculty at SBB Ilyas Appasaheb Jedi College, Pune, Maharashtra. Ma'am has done her PhD from Dr. Babasaheb Ambedkar University. With a lot of experiences, she is the founder of science faculty in the college since 2004. She has also done lot many projects at PG level. Madam has a specialization in actinomycetes culture and also have lot many research papers nationally and internationally, including 3 books. Her patent on sewage treatment is also been internationally filed, which result is going to be published by this month of the year. She has also been lifetime member of many microbiology association and also a member of Microbiology Society India since a long time. So it is very less to describe ma'am in such a short period of time. So today's lecture is on actinomycetes as a potential bioinoculant. I hope everyone is excited to hear from ma'am. So without any delay, I think we should get started to today's session. Ma'am, over to you. Ma'am, you can share the slides now.
Am I audible?
Yes,
Am I
ma'am.
audible?
Yes, ma'am.
Yes, ma'am, you're audible.
Am I audible, madam?
Yes, ma'am, you can continue. You're audible.
Hello. हेलो मैम, वॉइस नहीं आ रही आपकी।
Am I audible? Am I audible?
यस मैम, नाउ यू आर ऑडिबल।
Yes, yes. Sorry for waiting. I could not join from my computer, so quite late. I am very much thankful to Microbiology Society India for giving me this opportunity. Now I am going to present my research work. I did from 2012 to about 2019, almost 7 years. Ma'am, I want to share my screen. Have you done this? Please. Hello, sir. Please make me host so that I can share my screen. Hello, ma'am. Am I audible?
Yes, ma'am.
Yes, ma'am.
Okay, sir, can you please make me host so that I can share my screen? Prathamesh, sir, are you there?
मैम सईदा तमन्ना मैम इज द होस्ट, so she can make you only.
Okay. Tamanna, ma'am. Sir, can you just call her? Prathamesh, sir. She should make me host, then only.
Ma'am, I did. You are a host.
Okay, okay. My screen is visible, I hope.
Yes, ma'am.
Okay. So my topic of research was studies on rhizosphere actinomycetes and their use as potential inoculants. My research guide was Dr. V.S. Hamde sir, Professor, Department of Microbiology, Yogeshwari Mahavidyalay, Ambezogai. Now, starting from the introduction, I will just take 2 to 3 minutes just for introduction of my topic. As we all know, plant growth promoting rhizobacteria, they are all found in rhizosphere and rhizoplane. They colonize this area and they help the plant to improve the plant growth when artificially inoculated onto seeds or into soil. So there are a number of the different properties of these plant growth promoting bacteria. Those help the plant for improving growth. Among them, 5 of them were selected by me. Those were nitrogen fixation, mineral solubilization, hormone production, chlorophore production, and biocontrol activity. So actually, when we see the plant growth promoting properties, these microorganisms, they help the plant in number of ways. And this mechanism is quite complex. It includes number of the things like release of some biocontrol agents that somehow is going to help indirectly for plant growth promotion by the bacteria to the plants. Now, there are number of the microorganisms which were used as PGPR. Various groups of microorganisms have been used as PGPR, like Azotobacter, Rhizobium, Azospirillum, Pseudomonas, Bacillus. Also, Streptomyces. Initially, it was studied as biocontrol agent, but from 2012 in Kyrgyzstan, they started using Streptomyces fumanus as a by a plant growth promoting agent instead of the biocontrol agent. Now, as we know, there are different characteristics of actinomycetes which make them unique organisms when we compare them with other bacteria. They are free living, they are found in almost all environments, they have distinct earthy odor, they are gram positive filamentous bacteria, also they have the high G plus C content, they are spore forming and producers of number of secondary metabolites. When I did literature survey, I did the literature survey and also some site survey in the area of the Hadapsar near Pune region, Fursungi and Mandri. What I found is that the soil of this region was alkaline and the pH was ranging from about 7.32 to 8.52. Also, there were some literature I found which was supporting the thing that this area was having a very alkaline pH, and whatever fertilizers were used by the farmers, they were mostly the chemical fertilizers, and some farmers were using biofertilizers, but they were saying that they are not of use, as the organisms which were used mostly as fertilizer, they were having their optimum pH of 7, and the area was having the pH of about 7.32 to 8.52. So I searched for some other literature, and according to Gopal Krishnan et al., PGP potential of Streptomyces was reported on cereals and such as wheat. Also, there were some literature showing the Streptomyces as useful PGP organisms. So keeping this in mind, the aims and objectives were set by us. They were like isolation of actinomycetes from rhizosphere soil of various plants, screening of the isolates for various PGP features, identification of IA and siderophore by TLC and HPLC, effect of physicochemical parameters on PGP properties, micronutrient analysis of treated plant using ICP spectroscopy, pot trials using selective isolates based on mostly the multiple PGP properties. Then some of the promising isolates were chosen, and from those solid formulation was prepared. It was also assessed for its safety by using Ames test, fish toxicity and soil toxicity testing, and the field trials were carried out. So this was the framework of my research work. I have collected the samples from the rhizosphere soil of different plants. I have isolated about 100 different isolates, and among them almost 30 were taken for the screening of the PGP properties. And those isolates, about 5 isolates, I found they were having very good PGP properties qualitatively as quantitatively. So I chose them for pot trials. I made the talcum powder formulation, fish toxicity testing carried out, and carcinogenicity, and then we carried the field assay. So this was the total framework of our research work. Am I audible, madam?
Yes, ma'am, audible. You can continue.
Yeah. So when talking about the material and methods, we have taken the soil sample from rhizosphere soil of the plants, and this was the Google location from where we collected the soil from Kalepadal, Hadapsar and Urlikanchan, and we collected the sample from about 0 to 15 centimeter depth of the fields plots. The plants were hibiscus rosa sinensis, aloe vera. So some almost number of the plants in this list you will see they were the medicinal plants, and also we selected some plants near brick factory, and from the rhizosphere soil of this brick factory plant near plants near brick factory, we also collected the soil samples. So how we isolated these actinomycetes? These samples are the organisms where they were named, like say for example H, that means the soil sample collected from the hibiscus. Like that we named about 30 to 32 isolates, and we carried out the method of isolation as per described by the Kasimurugan in 2012. Serial dilutions of rhizosphere soil samples were carried out. Spread plate technique was used here, and the agar used was actinomycete isolation agar ready made from high media, and also the casein starch agar. Those plates were incubated at room temperature for 7 days, and the powdery dry colonies were selected and they were transferred to the fresh agar medium and stored for 4 degrees Celsius. The isolates were identified by performing the biochemical tests and referring to the Burgess Manual of Determinative Bacteriology. Then they were screened for the plant growth promoting properties, and the properties selected were indole acetic acid production, phosphate and zinc solubilization, siderophore production, anti-phytopathogenic activity, growth on nitrogen-free media, and some potent isolates were then sequenced by using 16S RNA sequencing. Phosphate solubilization. For phosphate solubilization, we use the method of Picowaskis, and that was used by some modification by Suleiman et al. In 2018. So we modified the CSA broth and the broth was incubated at room temperature on shaker for 8 to 10 days. After incubation, the broth was streaked on CSA plates incubated at 28 to 30 degrees Celsius. And then we observed the zone of clearance. Quantitative estimation of phosphate solubilization was carried out by using the method of Murphy and Relay as from 1962 with some modifications which has been used by Jain et al. In 2017. So that method was used for quantitative determination of phosphate solubilization. That was spectrophotometric method. We also checked the isolates for zinc solubilization as per method described by Sharma et al. In 2012. Trace minimal media with 0.15% zinc sulfate and isolates were added on this media, and they were incubated at 30 degrees Celsius for 3 days, and after that the solubilization zones were observed. Screening of eye producing actinomycetes. For this, the isolates were grown on tryptophan yeast extract agar, transferred to 5 ml tryptone yeast extract broth, and the method as per Bano and Musrat research paper in 2003 we carried out, and standard IA used was of high media. Detection and identification of indole acetic acid that was carried out by using thin layer chromatography as described by Barucha et al. In 2013, where we used the solvent system of N-butanol ammonia water, and we did the chromatograph, and that was developed by using Salkovsky's reagent. HPLC was carried out as per method described by Hariharan in 2014, and here we use the column C18 column and the solvent system of the acetonitrile water system containing 0.1% trifluoroacetic acid, and runtime was kept for of about 30 minutes. Mobile phase was methanol and water, and extract used was ethyl acetate extract. The isolates were then screened for the production of siderophore. As we know, siderophores are the iron chelating compounds, and thus they starve mainly the fungal pathogen, and they help the plant for getting their nutrients. So that's why we did the screening of these isolates for siderophore production. About 33 actinobacterial isolates were separately inoculated in succinate media, and they were incubated for 30 degrees Celsius at 30 degrees Celsius for 3 to 5 days on rotary shaker at RPM 200. Centrifugation was carried out at 10,000 RPM for 20 minutes, and the collected supernatant was used for universal CAS assay or universal chemical assay. We call it as a CAS assay for siderophore detection. Also, the qualitative and quantitative analysis was carried out as per method described by Piney in 1994. So in this method, the cork borer wells were bored on the CAS agar, and about 75 microliter culture supernatant of each isolate was added to the supernatant well, and then they were incubated at room temperature for 2 to 3 hours, and the color change from blue to orange, yellow, purple that was detected for the presence of siderophore. Then the quantitative estimation of siderophore was carried out by CAS shuttle assay as described by Piney in 1994, where the percent decolorization was calculated as per the formula which has been shown on this slide. Quantitative estimation of siderophore was carried out by one more method, that was the iron perchlorate assay as described by Atkin et al. In 1970, where we have used to estimate, we have did this assay to estimate the hydroxymate type of siderophore by using the ferrichrome as a standard. The standard were obtained from the high media. Then the siderophore was also detected by using thin layer chromatography as per described by Paul and Dubé. And also the HPLC was carried out, and the HPL reference for HPLC was taken from Shodhganga as per the procedure described by Bendale MS in 2014, where we used the UV visible detector, and the mobile phase was methanol and water, and flow rate adjusted was 1 ml per minute at 25 degrees Celsius at 220 nanometer. Then the pot assays were carried out first to determine the use of the isolates, where we prepared the siderophore rich broth, and this siderophore rich broth was inoculated with the seeds of the plant, and here we took mostly the chili and tomato plants, because they grow in a very little time period, so that we can get our results immediately for testing the efficiency of these isolates as PGPR in pot trials. So we carried out this experiment, and then we after this we took out the plants, and these plants were then dried. Then the weight of these plants, they were converted into the fine powder, and then they were sent for the micronutrient analysis. That micronutrient analysis was carried out by ICP spectrophotometry, ICP spectroscopy, and the plants were then dissected for observing the development of xylem and phloem. Then one of the important PGP properties that we see in all biofertilizer, the organisms which we use as a biofertilizer, that is mainly the growth on nitrogen-free media. So we also try to see whether these isolates are able to fix the atmospheric nitrogen or not. From that point of view, first we try to grow these organisms on nitrogen-free media, and the organisms which were positive or which were growing on this nitrogen free media, then we transferred them to the laboratory for search of NIF gene analysis. And this analysis of NIF gene was carried out by using the primers Paul F and Paul R as described in early research paper for NIF gene analysis of Streptomyces species in Dahal et al. 2017. Then in vitro screening for antifungal activity was carried out of about 32 isolates. Qualitative analysis of this antifungal activity was carried out by using agar-stick method, and for quantitative we carried out the dual culture plate method, where we calculated percent inhibition by using the formula which has been described by the Kumar Rajesh et al. Then seed germination assay of the isolates was carried out by using the method described by Mia et al. In 2012, where we calculated the percent emergence of the organisms by using the formula of number of emerged seedlings divided by number of seeds sown into 100. Pot assays were carried out using isolates, and the statistical analysis was done, where we used seeds of wheat, maize, chili, and tomato. And we also carried out the pot assay for taking the seed germination. Then the efficient isolates we found about, we screened about 32 isolates for PGP properties, and among them we selected 4 isolates depending on their best PGP properties. Those were A3, BF5, TU, and D. Then they were individually inoculated in 800 ml of the broth in 4 flasks, and then they were kept on rotary shaker to get final concentration of each isolate till of 0.8 to 10 into 10 raised to 8 CFU per ml. And by taking this, then we added about 1 kilogram of talc. This was added in 1 kilogram of talcum powder, 15 gram of calcium carbonate, and 10 gram of carboxymethyl cellulose that was allowed to mix in with 400 ml fully grown bacterial inoculum. The shelf life of bioformulation was calculated by using the serial dilution technique, and the samples were kept at room temperature of storage. Then the safety assays were carried out by using mostly the kits which we obtained from the BioEra Life Sciences, and as per method described on the kit, we used the organism Salmonella typhimurium for carcinogenicity testing. Also, we carried out the environmental toxicity testing of the biofertilizers, where we use the fish toxicity testing. As per OECD guidelines, these protocols were carried out: soil toxicity, fish toxicity, and then we use these samples for further field trials. So field trial was carried out as per method described by Patil et al. In 2012. And here we use the variety of maize and wheat. So the two plants were selected for field trial, and these are the Google locations where we carried out the field trial. In this field trial, we applied the 3 doses of our biofertilizer formulation on each plot of 5th, 25th, and 45th day after the seed sowing, and also except on the control plant, powder formulation that was mixed at 5 gram per plot, mixed with 250 gram of sterile soil, and that was applied to the plots. Then 10 plants from each plot were randomly selected for periodic observation of different growth parameters. So now moving towards result and discussion, we found about number 32 isolates actually we were selected, and about 100 initial isolates were there. So they were named according to the source from where they have been obtained. So the isolate A to D, where that name was given to the isolates which were isolated from wheat, like that brinjal BR1, BR3, like that. See, this was the just naming of the isolates, and the isolates which we obtained from the plants near brick factory, that were named as BF1 to BF12. So these are some of the isolates of our research work. These are the isolates specifically from the rhizosphere of medicinal plants. Then these are the isolates from the rhizosphere, mainly the plants which were near to the big brick factory. So this is the chart of biochemical characterization, where we did all the biochemical tests as per Burgess manure, gelatin hydrolysis, starch hydrolysis, oxidase, H2S production, catalase test. So these are the results of all the isolates. And the some of the potent isolates, we did 16 srRNA sequencing. So 5 promising isolates which we sequenced by 16S rRNA, and we found that the BF5 was Streptomyces, showing 99% similarity with the Streptomyces parvulus strain NBRC. A3 was showing the similarity with Streptomyces flavulus strain NRRL. Then A2 was showing the similarity with the Streptomyces flavifungini strain. That was the strain number BB1. Then there was the D, Streptomyces sp PF9. TU, that was Streptomyces griseus strain A20-1. So the isolates, when they were screened for phosphate solubilization, you can see in the slide also, and also I have kept one particular plate here. So you can see here such a clear phosphate solubilization was observed, and the efficient isolate BF5, it has shown the maximum phosphate solubilization index that was in between, that was more than 8. So this is the graphical representation of phosphate solubilization zones and the phosphate solubilization index that we did for about the 33 isolates. So then we also studied the effect of different parameters. So effect of pH on phosphate solubilization and the effect of the temperature and phosphate solubilization. So the maximum phosphate solubilization that was in the range of mostly in the room temperature range, and the less phosphate solubilization as there is increase in temperature. Also pH, it was found that these organisms were able to work at higher pH also. Then the isolates screen for the zinc solubilization, and this is the result of zinc solubilization. So here also the isolates like isolate D, BF5, they have shown the best zones for solubilization. Mostly the plants which we selected near brick factory, they gave the best result for the zinc solubilization. The isolates were also screened for the IA production, and maximum IA production as per this A3, the organism A3, it shown the maximum IA production. Maximum IA production was found. So again, just there was some issue, so I am sharing my screen. There is some issue for the slides. Am I audible?
Yes, ma'am.
Just a minute. Something has happened, so Sir, can I send you my PPT so that you can? Prathamesh sir. Prathamesh sir. Prathamesh sir. Are you there? Am I audible, madam?
Yes, you are audible.
So, madam, can I give you my PPT so that because it is not working from here, I don't know what is the problem.
Okay, we can move into the question answer session now. You can share in our group itself.
Hmm?
You can share in our microbiology society groups.
Okay.
I think we should move into the question answer round.
Okay.
The participants, anyone have any question?
Just a minute.
Ma'am is open for discussion and question answer round. So, do anyone have any question?
Yeah,
Ma'am, are you available?
this is Dr. Aruna from Hyderabad. Can I ask question? Can I ask my doubt?
Sure.
Yeah. So, ma'am, what is the purpose of using this talcum powder? And then, what is the role actually? Its function? You're adding talcum powder, right? I just want to know the function of it. What purpose actually? What is its role? Hello? Am I audible? It's not there in the session. Speaker. Sunita ma'am? I think she is not there in the session. We left the mute. I'm just saying. Yeah, I'll mute myself. Tamanna ma'am, I'll mute myself because I'm not getting answer from Sunita ma'am.
Yes, ma'am, you mute. Once he comes, I will let you know.
Yeah.
Am I audible?
Yes, ma'am.
Okay. So, in short, after the studying all the characteristics, when we did the pot trials, we found that there was increase in micronutrient content also of the treated plants. And when we did the field trial, we found the results were significant. There was increase in the growth parameters of the plants, not only of wheat, tomato, but almost all the different plants, they showed the increase in the growth percentage. So there was not only the qualitative increase, but quantitative increase was also there in case of the treated plants. So when we did all the toxicity testing, it was the biofertilizers were found to be free from all the types of the toxins. And so they were the promising isolates for carrying out the further experiments. Also, the results of the pot trial of mostly the treated plants of the tomato, and they showed that untreated plants xylem and phloem was not well developed, but the xylem phloem development was also early in case of the treated plants with our isolates. Almost we studied different parameters like copper, then iron, manganese, and zinc, and we found that when we compare with the control group of the chili, there was about the 90% increase in the amount of copper, iron uptake, and manganese uptake. So
Ma'am, you had some questions.
yeah, okay.
So, ma'am, Sandhya Rani ma'am asked that, can you tell about the biochemical reaction of actinomycetes?
What?
Can you tell
Yeah,
about the biochemical reactions of actinomycetes?
biochemical reactions in the sense what you are asking? I'm not getting. Biochemistry of the production of different components, you are saying?
Ma'am, is Sandhya Rani ma'am available here? Hello, is Sandhya Rani ma'am available? No, I think so. It's not available to clear the question. So, you have
Yeah.
another question. Why are you using talcum powder?
Yeah, that is used as a carrier base. We used it as a carrier base. Because
Another
that
have
is
is
cheap carrier base which we can use for the formulation. So that's why we use the talcum powder.
Any brand can be used, ma'am. This is Dr. Aruna from Hyderabad. So this is
Yeah.
regarding talcum powder only. So any brand, like any company, powder can be used. Is there any specific powder is
So there was a, actually we use the sandal powder.
Ponds? Ponds. Is it Ponds?
Yeah.
Ponds are the-
Yeah, pond
Well.
sandal. Mhm.
Okay, right. Thank you, ma'am.
Okay. Actually, I am having a lot of results, but there was some problem, so I could not show you
Yeah.
the promising results of my research work.
It's like it's a very good information, good data.
Yeah,
you
I have also deposited the cultures of my research work in NCMR.
Okay.
3 cultures of my research work I have deposited in NCMR. Those were mostly the streptomyces. Now, normally what we think is the streptomyces are used mostly as the biocontrol agent. Yes, for biochemical identification, mostly my aim was to screen mostly the organisms for plant growth promoting properties. So I did the enzymatic analysis also, like enzymes which support the growth, like amylase, which supports the growth mostly for the seed germination. So I did the biochemical test of amylase production, also H2S gelatin. I have named gelatin hydrolysis, H2S production. So there are number of the tests as per described in the Burgess determinative bacteriology. So I carried out all, and mostly the yes. Any question?
மாடியில இருந்துச்சு அது துணி அள்ளிட்டு இருக்காங்க. எங்கடா அண்ணன் வழியாங்கன்னு மேல கொண்டு போட்டு ஒண்ணுதான் முடிச்சிருக்கேன், கூட்டியாச்சு. இங்க நடுவுல ஒட்டடிச்சு கூட்டி.
Any other question?
நோ பாயிண்ட் அவுட் நான் வந்து 3 அலசிட்டு அந்த போர்வை அரசதான் வந்தானா.
Also the research work was presented at AMI and also at the
தலைய போய் பாரு கண்ணாடியில,
at international
போய்
and
அப்படியே பாரு,
national.
தேய்க்காம இதுல பாரு. அப்பாங்க எங்க போச்சுன்னு தெரியல, காணும். அங்க போய் மோட்டர் ஃபிட் பண்றா.
Once audio is work there which is disturbing, that should be. Is there any other question?
Ma'am, there is one more question. Tell us about the formulation of bioinoculant. What was CFU per ml?
That was about 0.8 to 10 into 10 raised to 9 cells per gram. That was the formulation. That means CFU. I am telling about CFU, colony forming units, because we see it in the form of the colony on the plate. Specific biochemical test, see what I did is I directly use the selective media like Actinomycetes isolation, agar and CSA. And then the important tests for this Actinomycetes are mainly the tyrosinase, mostly the enzyme production. There are number of the tests. And finally, as we did the 16S rRNA sequencing, then we get to know that these are the Streptomyces, different species of Streptomyces. Only 5 PGP properties we screened. Those were IA production, siderophore production, and also the experiments which I carried out, those were also with the siderophore rich broth for pot experiments. And for field trial, we use the particular solid formulation. So initially the trials were with the isolates, and finally after preparation of the biofertilizer, the trials were with the particular biofertilizer formulation, solid formulation.
Ma'am, fish toxicity is you're checking the toxicity against aquatic organisms. Is it
Yes,
like that?
yeah. OECD, that is necessary as per OECD guidelines. It was necessary to check the because that fertilizer is going to finally go in the soil and water. So the soil toxicity and the fish toxicity testing was carried out, and for that I used the kits of BioEra, and by using those kits I did the toxicity testing also.
So you got the fish of different
Yeah, guppy
different
fish.
types, and you made it in the lab, and then you have added your thing bio.
No,
it like
that no, actually I am having one this what you say fishery near my home in Hadapsar.
Okay.
So I took some area from them and I did the experiment there, because we have to maintain also that guppy fish that were used for this experiment. Those were common name of those fish was guppy fish. So there is one fishery in Hadapsar. So I did the experiments there. Actually, this work was of about 7 years I required for this.
Yeah, it's a good work, ma'am, and you have included many parameters. Nice.
Yeah, actually results are also very good, but because of some issues, sorry, I could not do this. Mm. Also, I presented my this research work in AMI conference and in VSI Pune when there was Narendra Modi for this conference in Pune in 2016. So that was also very good opportunity for me. I explained my research work in front of PM Modi sir.
Have you published your paper, any research articles, ma'am?
Yeah, sir. I have about 7 to 8 publications.
Good.
One of them is in Web of Science and about in UGC approved journals. I have published the papers.
Congratulations, ma'am. Good work.
Screening of this one international journal of recent scientific research is there. Then one of them is International Journal of Current Microbiology and Applied Science, that is there. So like that about in one I published in Springer also. There is one chapter in Springer also. So in short, I would like to conclude this research work as what I found in all my results, that BF5 isolate, that was the one of the species of Streptomyces, it increased shoot weight of sugar cane. I did experiments on sugar cane also, and that was up to 95% when compared with the control shoot weight. And we know that the sugar cane is mostly used for the production of juice. So if shoot weight is more, then there is importance of this sugar cane. So the variety I used was the Nira variety of the sugar cane, and it also increased shoot weight, height of the plant. It improved the height of the plant in case of wheat, and also it showed the presence of NIF gene production. That was very important finding of my research work. BF5 isolate, only one isolate I found which was showing the nitrogen fixing property, that was Streptomyces parvulus. And when I did the NIF gene analysis, I found that the mostly the NIF gene was not from actually the Streptomyces, but it was transferred by horizontal transfer from the other organism, Enterobacteriaceae. So that was plasmid borne gene. So that's why the BF5 isolate, which was isolated from the plant's near brick factory, rhizosphere of the uh, plants, which were near brick factory. So that's why I think there was the horizontal transfer, and the NIF gene analysis was very promising in my case. Also the tomato and chili plant, when I inoculated the siderophore rich broth in the pots, and when I did the micronutrient analysis, there was also the significant increase in the plant parameters. Also they have very good effect on the number of the cob of maize. I did the experiment on wheat, maize. Also the cultures I deposited in NCMR, and they have got the accession number of MCC 3808, MCC 3809, and the isolate A3 has got the accession number of MCC 3951. So nowadays what we say is the research should be the applicative, and it should be also made public. So from that point of view, I have deposited the promising cultures of mine in the NCMR. And the step next, I am doing the research work specifically on Streptomyces parvulus, because what the isolate what I got, that was also fixing nitrogen along with all plant growth promoting properties. So that must be checked again for the interaction of the plant and the microbes.
Sugarcane experiment it was like pot culture or it was a field culture, field experiment?
No,
Sugarcane
sugarcane I did for about 2 months, pot culture.
pot, okay.
Yeah, sugarcane was for pot culture. Wheat and maize I did for field trials.
Okay, field.
Actually, because sugarcane took a lot of time on the field.
Correct.
It requires almost 2 years, madam, for detailed study at field level. So I did. One of the
congratulations from my side. Very good work. Very good information. Informative work, ma'am. Thank you. Thank you.
Thank you. Thank you very much.
Thank you, ma'am. It was a great lecture, and your research work is really very interesting. I'm thankful that you shared your work from A to Z, showing many parameters. And if a new researcher is planning to work, then it can easily design their work.
They can contact me.
Yeah. They can easily design along with the methodology part where you included biochemical analysis, and also very nicely statistic analysis has been done. So thank you so much, ma'am. Lastly, I would like to thank Desmukh sir for giving me this opportunity to host today's lecture. And I'm thankful to everyone who have participated. And ma'am, you can join our group if someone wants to connect. Then they can meet you there. Many of them wanted to look into your research papers, also the published papers. So it will be a great interaction there. So with this, I would like to end today's session. Thank you so much, everyone.
Okay, they can contact me on personal WhatsApp also, where I will share my papers. Whoever wants my research papers.
Ma'am, can you please share your contact number or mail ID in the chat box so that I'll.
Yeah. Just a minute.
Yeah.
Just a minute.
Correct. Thank you. Contact number and then mail ID. Yeah. Both.
So I've shared. Can you see, madam?
Yeah, that's what I'm checking.
Yeah, Dr. Sunita Sakure @gmail.com.
Got it, ma'am.
And my cell number is 9325510030.
Yes, ma'am. Got it, ma'am. Any queries regarding if I'm planning this kind of experiment, please I'll be in touch with you.
Okay.
Yeah. Thank you, ma'am. Thank you. Thank you once again. Thank you for all the organizers of this session. Thank you.
I am also very thankful to Microbiology Society India for giving me this opportunity and sharing my research work. Thank you, A.M. Deshmukh sir. And also I am thankful to Tamanna madam and Prathamesh sir. But there was some technical difficulty. So I feel very sorry for that purpose.
Shall we exit, ma'am? Tamanna, ma'am? You can leave, right?
Yes.
Session is done for the day. She's not there in the session. Thank you.
Thank you.
Thank you. Thank you.